Welcome to our GSA webinar series on National Advisory Council Aging Priority Research Concepts. Today's program is on interorgan communication in aging. Please introduce yourself in the chat and know that our conversation is being recorded and will later be available on GSA's website. Please use the chat box as well to ask questions, and at the end of the presentation, our moderator will help answer these questions and give them to the appropriate panel members. And remember, if you like our format, please invite others to join us for future programs. Okay, next slide. Our program has been developed by a GSA work group. I am Jim Powers, the lead for the work group, but I'd also like to acknowledge my other GSA co-workers, Daniel Gann, Sam Lee, Meredith Troutman-Jordan, and Pan Saturis. Next slide. Today's presentation will be by a panel of program officers from the NIA, including NIA Division of Aging Biology members, John Williams, Amanda Boyce, Hongwei Go, Mulu Tulahan, and Gustavo Duke, who will be our chair and moderator. Dr. Duke is the chair of geriatrics at McGill University. And I will now turn the program over to Dr. Duke to introduce our speakers.
Thank you very much, Dr. Powers. It's a pleasure to be here. Thank you very much, GSA, NIA, for organizing this interesting activity and for inviting me to moderate the conversation today. One of the important things for to start with is just to ask you to be as interactive as possible. So please feel free to write your comments, questions on the chat. And we will try to approach them maybe at the end of the session. We are going to have plenty of time for discussion. So please feel free to write your comments and questions, and we will work on it as soon as we are done with the presentation. So as Dr. Powers mentioned, we have 4 presentations today. All our speakers are health science administrators at the NIA Division of Aging Biology. And I would like to start by inviting Dr. John Williams to give us the first presentation. John?
Thank you very much. So I think we have 2 speakers and then the rest of the panel to assist with answering questions. We are very excited about this this initiative. Excuse me. I personally think it's long overdue, and as you all know, that you've seen in the literature where there are a growing number of reports where a tissue-specific knockout has an effect in a non-target organ. And so there's a growing interest, at least, well, in parts of NIH and specifically within this division, to try to accelerate work in this area to grow a greater appreciation for the need to understand mechanisms of tissue crosstalk. And so this is widely focused on so in writing an application, obviously you have to convince reviewers that you have the expertise to do both ends of the tissue crosstalk, and so There is some limitation on the number of organs that you can include because, you know, it has to be, I'm not sure how. So it has to be feasible in the life course of a funding period. So, but it can be essentially any 2 organs, and then so I think I will stop talking and let Hongwei present some slides that summarize some of the key points of the RFA, and then hopefully we can have open it up to get all of your questions. I'm hoping that we can get everything clarified for you as we move forward. Hongwei?
Thanks, John. I'm Hongwei Gao, the program officer of NIA. I just want to give you a very brief overview for this RFA. I want to mention that although the human subject study are encouraged, however, this RFA doesn't allow the clinical trial studies. And this slide shows you some specific area of research interest include, but are not, of course, limited to these topics. For example, the changes with age or health status in communication between 2 or more organ systems, and the changes in interorgan communication that alter the apparent rate of aging, including the environmental exposures, and the changes in interorgan communication by interventions that have been shown to lower the apparent rate of aging. For example, the intervention that can extend the lifespan or health span, such as pharmaceuticals, diets, and/or exercise. Some other potential topics include the potential of regulating interorgan communication as an anti-aging strategy, age-related change in the activity and function of a tissue-resident immune cell in the context of mediating interorgan communication. For example, the tissue-resident T cell, B cell, neutrophil, macrophage, and the dendritic cell. And how age-related change in neural function alter peripheral organ physiology and function. This include sympathetic nerve system and also parasympathetic nerve system. Next slide, please. And this slide shows you some key aspect of this funding mechanism. We are going to commit $5.8 million in the fiscal year 2024 to fund up to 10 awards. And the maximum project period is 5 years. There's only one receipt date, is June 1st this year. No resubmission is allowed. The direct cost per year is $400,000. We encourage application from multi-PI, and we plan to hold mini symposia, at least one mini symposia every 2 years. Next slide, please. So this slide shows you some important dates and the information here. If you have any questions, we strongly encourage you to talk to program officer. Thank you.
Thank you very much, Hongwei. So these are the details, specific details that we have in the RFA. Does Amanda or Mullen would like to add some information? Amanda?
Hi everyone. No, I'm happy to take any questions from the group. I mean, this is a kind of the central concept within the branch of aging physiology. So I'm really excited to see questions and the applications as they come in.
Mulu?
I'm Mulu Tlahun. I manage DAB's immunology portfolio as well as the microbiome, currently microbiome portfolio. And if you are interested to, considering any application related to immune aging, inflammatory aging, it's very broad range, or microbiome, you can contact me. And we have upcoming workshops on, areas related to this aging, as well as inflammatory disease and immune senescence in May. And check the website. And if you are, if you are interested, please, register, and you can get more information during that workshop, just to give you some, broad information.
Excellent, thank you. So we are starting to receive some questions from the participants. The first one, I think that this is something that John mentioned, but Linda asked some clarification regarding two organs, because it was mentioned two organs at the beginning, and Hongwei mentioned two, or the guidelines, two or more organs. So John and the rest of the panel.
I'm happy to clarify. I apologize. So it has to be at least two. It can be more, but it has to be at least two.
Okay, and John, continuing with that idea of more than two, or is how do you, in terms of the evaluation criteria, can innovative interactions be highly valued? I mean, organs that we don't think are going to be interacting, and the proposal try to interact them. How do you see this kind of approach?
So I think organs are highly interactive, and so the applicants are writing applications to an NIH review panel, an NIA review panel. So it will be aging focused, but applicants obviously have to convince the reviewers that they have preliminary data supporting their premise, and that they have the expertise to carry out the work. So, I mean, it's the normal caveats that you have in writing a grant. But if you have data showing that there's interaction between two or more organs, and that you're going to pursue that, then great.
Perfect. There is another question regarding the clinical trial. So the person who is asking, Rachel, says that there was information about clinical trials allowed in the first slide, but it was just mentioned that RCT are not for this RFA. Could you please clarify that? And maybe Hongwei?
The clinical trial is not allowed for this RFA, but we encourage to use human samples or any kind of a human subject related study. Yeah.
Okay. Anybody from the panel would like to add in that sense, or in the previous question?
I would like to second what John says. Obviously, what you're going to come up against in peer review is, if you are talking about multiple tissues, is that you have the expertise in the multiple tissues. So it's probably going to require a team approach, though I know that there are some people with a long history of already sort of looking at interorgan communication. And I actually asked John to comment. So one of the things that we've talked about behind the scenes is what happens if we get 5 grants that are studying muscle communicating with bone. And so, John, what happens if we get 5 grants on one topic? What would you like to see?
So we mentioned in the RFA that we're looking to build a consortium of diverse applications, and so I would be hard pressed to be able to justify funding more than 2 grants on the same organ pair, and it would be only in light of them looking at I don't want 2 grants doing the same thing. And so that would it would need to be a different approach, different topic, different signaling along those lines. But the goal is to have a consortium, and that the meeting that we anticipate having in years 2 and 4, I think, since basically year 1 you're gearing up, would be to have people come together, and I anticipate that there will be crosstalk between grants, essentially, because it's not just one organ talking to one other organ. The one thing that is clear from MotorPack data is that, for example, all organs respond to exercise. It's not just muscle, it's not just fat. Everything responds, and if everything is responding, then in order to maintain homeostasis, there's feed forward, there's feedback. It's a highly complex, orchestrated interaction of organs that need to stay happy. And so I think we will find that there is a lot more interaction than people are currently thinking about.
There is actually a question here associated with what we discussed before about people, I mean, our applicants looking at novel organ interactions. And the question from Candice Cham is, would the gut microbiome be considered an organ?
I'm not sure I would consider it an organ, but it clearly affects other organs. And so in that context, it seems appropriate.
Any other comments from Mulu, Amanda, or Hongwei? Okay.
I think I also agree what John said. I mean, it should be considered as, even if it's not organ, it should be considered, since it's affecting multiple, for example, especially the immune system. It should be considered as one, and considering microbiome and other immune response, for example, can be an example.
Yeah, that's a good example.
Mhm.
Chris Hernandez asks, can you comment on differences in expectation for the U01 mechanism versus the R01 mechanism?
So the U mechanism denotes that it is a cooperative agreement. So a simplistic explanation is that there's more NIH involvement, so you have to listen to me more than you might normally want to, and so that it's really, I'm joking. It's more about encouraging grants to be open-minded about the potential crosstalk between. So you're doing bone and muscle. Well, what's going on in the kidney? What's going on in the liver? And I think we will find that some signaling mechanisms that are occurring in your organ pair may also be affecting another organ pair minimally. And so I think there's a lot of opportunity to enrich the science by broadening the scope through the use of this cooperative agreement. So it's like a fancy R01.
So yeah, I want to add to John's comment. Each award will will be assigned a scientific officer. So that person will coordinate the communication between different awardees. Like John said, probably share some new technology, a new advance from different awardees. So.
Thank you. So we have a question here from Paul Cohn. So can we propose human clinical studies that are not randomized controlled trials with biopsies, novel sampling methodologies, etc., that interrogate IOC in response to acute stressors, exercise, feeding, fasting, etc.? Who wants to comment about this, about the randomized controlled trials and samples?
So it sounds like it. An Ames page would be helpful. We would. So what I don't know is this more clinical outcomes, or is this a mechanistic study using human tissues? And if it's the latter, then yes, it's appropriate. But if it's clinical outcomes, then no, it wouldn't be appropriate.
Okay. Minghui Zhu asks, does this RFA have interest in cardiovascular aging?
Sure, any organ system.
Any organ systems. Linda Bonwell asks, should this U01 be more multi-investigator than a single investigator R01? Preferably. Okay.
Because, well, so our feeling is that in this day and age, multi-PI awards are much more common than they used to be, and given the multidisciplinary nature of the science, they're often essential to fully develop an area of investigation. So the multi-PI is encouraged. I mean, there are people that that are established enough that they can make the case that they do it themselves, but it's a calculus that each PI needs to make as to whether or not they can convince reviewers that they can do this area of crosstalk that they're proposing.
Thank you. Any other comments from the panel? This one, this question from Indira Misoreka. How would you review panels? How would review panels be instructed to evaluate cross-organ signs? Often there are tissue-specific aspects that need to be considered. Anybody from the panel would like to answer to that, Mure?
So we will discuss with the reviewers that are identified the goals of the RFA, and there's so they, you know, so we will highlight what has been presented in the RFA and what we are trying to achieve. There, I would hope they would listen to us.
I mean, we'll work closely with the SRO. Hopefully,
Yeah.
I mean, it depends on who's assigned. They generally will ask us for recommendations for reviewers, or at least topics that need to be considered. And it's really going to depend on what the application pool ultimately looks like, right? So we will try to make sure that we cover as many organs as possible, you know, knowing that we may not hit them all, but we'll probably hit something at least peripheral to, at least your organ system. So we will do our best to cover it. And then there will be reviewer training where we will, it is an RFA, really focus on what the RFA is asking for and expectations for reviewers. And, you've all pretty much on here been reviewers. You try to respect what the RFA is, the kind of gestalt of the RFA.
Thank you. Any other comments regarding that question? So Hongwei just explained about the letter of intent, about the information, the contact information to submit your letter of intent to John. There is a question from Jeffrey Mason. Can a proposal be gender specific, focus on, include only one sex?
If there is a scientific justification for only one sex, prostate cancer, breast cancer, well, not so much breast cancer, you would have to justify the focus on one sex or the other. But if you're something ovarian related, obviously is going to be female. But if it is true that there are a lot of sex differences, and so if you're going to propose one, looking at only one, you have to have a scientific justification for not looking at the other.
Any other comments? Okay, I have a question here from about, we have people from international, GSA has a wide international impact, and we have some people interested on it from other countries. Some of them, I recognize the names here as young investigators. Could you please, any of you guys, explain a little bit about the role of the foreign component on these grants, and, give some guidance to the USA-based investigators on how to partner with these international potential co-applicants?
So there is an added level of review which occurs at council if the contact PI is in another country. And the council focus is on whether it is appropriate to be funding a grant in another country when there are plenty of people, for example, in the States that are doing similar sorts of work. It's dicier for a PI from a foreign country. They can be co-investigators on a grant where they are contributing something, but it's a bit more of a hurdle for the PI to be in another country, because it has to be, I believe, the gold standard that we hear when I hear it being justified at council, because it does happen. So, but they're bringing something unique that is not being done here. No, and I know there are some European studies, for example, that can do science that typically isn't done here. So as I mentioned before, an Ames page helps. But there's, I think the more common approach is for them to be on someone else's grant that is a PI in the States. But that's not to say it's not possible. But again, talk to us. And an Ames page helps a lot.
So just so you know, for this particular announcement, though, foreign applicants aren't eligible, but foreign components are allowed.
Yeah.
So, and I see Marco followed up. I think that means you cannot be a PI, correct, John? Do you know if MPIs, if a non-contact PI is a foreign component?
So I think that's I think or at least I thought the foreign component was fine, but it wasn't the PI.
Yeah, Marco, will you follow up with us? We'll loop in grants management to make sure things are a little different with MPIs, you know, where it's the contact institution and there's an issue with how the funds are distributed, those sorts of things. But we can follow up offline.
And that's just not to sound very discouraging to the international audience that is joining us at this moment, please, including myself. I just want to make sure that some of these crosstalk interactions have been already developed amongst international organizations and investigators in the USA. What I want to say is that some of our investigators or your investigators in the USA have already established links and preference to use to share models. You know, they have probably, there are models that could be already available in the States, but you know, they already have the confidence, the preliminary data, probably several publications sharing that model. So I just would like to encourage, and you agree with me, to encourage those attendees here in the future to discuss it with their collaborators in the States to become their PI and then apply together. What do you think, guys? Is that correct?
So there was a lot there. I think it I think I need to see it on paper and okay, who is doing what. But being a multi-PI is fine. I think the foreign component needs to be bringing something unique to the grant, and that's generally what council is looking for. Because it goes through the regular scientific review. The reviewers say this is brilliant, give it a great score. The added level occurs at the council meeting when council members have been asked to review it because it has a foreign component. I don't recall a grant that had a foreign component being turned down because of the foreign component. And so, but typically what the council members ask for is, you know, what are they bringing to the table that we don't see elsewhere or something like that. So it is clearly possible, but talk to us.
Yeah, okay. I think that's a good point, and that probably clarifies what some of the, because you know, I'm highlighting the international nature of our GSA, right, and we are attracting lots of interest from other countries.
Okay,
that's in summary what I wanted to highlight, John. The other question comes from Marco Brotto. He says, in line with a previous question, there is still resistance or this common question, or where is the crosstalk coming from? How important for this RFA is the specific identification of the mediators of the crosstalk?
So it doesn't matter what organ is the originator of the signal. I would say that in order to convince a review panel that your project has legs, you would need to have some preliminary data identifying, I mean, it's possible to sell a project based on the strength of the preliminary data that you have clear crosstalk. I think it would be a harder sell if you didn't know what the mediator was, because the description of the study tends to become a little too open-ended for a lot of reviewers. And so I think the more competitive applications will have a mediator identified, and then they're trying to determine the nature of the crosstalk
And
the mechanisms of the crosstalk.
yeah, I think this is a question related to mechanism. We not only want to see the change related to the interorgan communication during aging. We want to see the mechanism underlying it and how it may affect the organ dysfunction or aging.
Excellent. Ming Hui asked about, do you consider fat as an organ? Does this RFA have interest in crosstalk of fat or fats with other organs, such as vasculature in a specific organ like liver or heart?
The short answer is yes. From my time in science, all of the fat depots that have been identified that I know of are all metabolically distinct. And so while I would hesitate to call fat an organ, it's kind of similar, at least conceptually, to how we're envisioning the microbiome. It affects a lot of different organs, and there's resident adipocytes, that clearly affect other organ functions. So yes, we are interested.
Okay.
But I mean, it does have the same caveat of convincing the reviewers that you have the data that supports the premise, and it's strong enough to convince reviewers that the work needs to be done.
Link with that, John, there are multiple ways of communication and crosstalk. Are we talking, we could go from, a specific letter addressed to the other organ, or it could go from smoke signals or whatever. My question is, how are we going to define how specific, or how do you think that a specific communication should be? Because sometimes there are factors that are solved in the serum which reach other organs, which doesn't mean necessarily that is addressed from one organ to the other. Could you please, that's in the main concept of communication and crosstalk between organs.
I agree, but you're writing an NIH grant, and so you need to be able to convince the reviewers that it is not too open-ended. I mean, reviewers typically are looking for a defined project, and if you can't, I think you have to have a way of making it a defined project, and if you can't say this is so is the goal to identify the organ. Again, I think it's one of those sorts of questions that, I mean, there are lots of endocrine mediators that circulate, and we don't know what the what the potential end effects. So if you look at the hypothalamic factors, it used to be that the literature thought that they were just signaling in one place. Well, it turns out that for that there are multiple. If you now do RNA-seq, you can find hormone receptors in organs that weren't previously considered to be targets. And so, if the protein is there, then chances are it's doing something. So I would say you need to be able to build a solid enough story that reviewers are going to believe you have a signaling pathway that is intact, and that you are pursuing the mechanisms by which it's acting.
Thank you.
And Ames page helps.
Yeah. Any other comments?
Yeah, I just wanted that last statement that John made, not the Ames page, that's helpful, but this really is focused on mechanism. So it's really hard to do that if you don't have a proposed mechanism, right? So rather than if you've started with a wide open question, it's really nice to have a really focused and there may be an intermediary tissue that have to go through the liver to get to the heart or whatever it happens to be. So yeah, but it has to be pretty, when you write the R1, you have to have the preliminary data to point us in the right direction and then spend the time trying to really dig down into the mechanism.
I like that you mentioned about the intermediate organ, Amanda, because that involves, it doesn't have to have direct communication. It could be, it could have an intermediate, which is nice. There is a methodological question here. My question is, this is from Shen. My question is also related to U01 versus R01. When we prepare U01 grant application, do we need to add anything in particular for this mechanism compared with regular R01? Thanks.
So I haven't actually written in a U01. So I'm not sure if the form is different. It is implied that it is a cooperative agreement. So as I mentioned earlier, there is a larger presence of NIH essentially providing information to all of the funded investigators and encouraging interactions between groups. I am not certain if the U01 application package asks you for something different than what an R01 does.
I don't think it's hugely different. In this particular one, sometimes they will have, the expectation here is that you are willing to meet at least twice to have a meeting. Oftentimes there is some stuff that's buried in the U01 language where there are expectations for you to do something. In this case, it's more that you are, it's sort of passive. You'll read it if you go down to, section 4, I think. It talks about what it means to be a cooperative agreement and that the expectations, but nothing specific in the application that's needed.
Okay, thank you. There is a question here from A1. Does this RFA have interest in how another organ system, for instance, muscle aging, affects the progression of Alzheimer's disease?
So the short answer is yes. And it's interesting, so there are multiple organs deterioration that do track with Alzheimer's. Sarcopenia tracks, osteoporosis tracks, but getting to the mechanism is the trickier part, and so I would say yes, there is an interest if the again, it's one of those things, and we actually have a program officer from our division of neuroscience that is listed on the RFA. But if you send us an Ames page to get input, I can tell you fairly quickly. So the rough description would be that if the target is in the brain, then it will go to our division of neuroscience. If it's a mechanistic study on the peripheral organ, but it has elements that relate to Alzheimer's, then probably come to us.
So I want to add it. So if it's a total focus is on Alzheimer's disease, I think is not a response. You have to focus on the communication between different organs. Yeah.
Indira asked a question that I think is very interesting because, you know, about the translational component a little bit. Would therapeutic interventions that affect the same pathology in 2 or more organs be considered?
Would you repeat that, please?
Would the therapeutic interventions that affect the same pathology in 2 or more organs, are they considered? Would they be considered?
So we're talking for so a drug affecting multiple organs. Is that what we're talking about?
For the, according to a question, it says affecting the same pathology in 2 or more organs. I'm not sure about the same pathology what refers to. Could be fibro. I don't know. Maybe Indira, you want to write it down
or something. I think it's, you know, if it's something like fibrosis affects multiple organs, and so it would be more interesting, or at least from our perspective, if you were addressing how you're getting the fibrosis in multiple organs. I'm not, so I would need an Ames page to help me out here.
Yeah, I'm thinking, I'm envisioning it, John, like, okay, let's just take estrogen because it's going to hit everything, right? Would an intervention of estrogen alter interorgan communication differently in, I guess, sorry, with aging, it's a little harder with that, or pick a metformin or some drug that you'd expect to hit multiple tissues. I can see how you could put it together, adding the additional complexity of the pharmacological intervention, but I think it's an issue of looking at Ames pages. Honestly, I think you're right, John.
And if I can link that question to something also in the interest of translational research, you know, is this this RFA encouraging mechanistic alone, or do you give any value or higher value or more value to those applications who have translational potential into future therapeutic application that target multiple organs and systems?
So I won't tell you exactly what I think.
Please.
I think things translate when they're ready. We are interested in the mechanisms by which the crosstalk occurs. Obviously, if there is translation potential, reviewers tend to like that. This is a basic mechanistic grant as far as I'm concerned. And nobody has ever translated anything in one funding period. And so I think it's inappropriate, frankly. But I understand reviewers' appreciate if something has translational potential. Frankly, I would not oversell it because reviewers do not have to agree with you that it has how translatable it is. And fundamentally, this is a mechanistic application.
Okay.
And
But this is a, go ahead.
Go ahead.
Sorry.
So yeah, thanks. So I want to add that the interorgan communication during aging is still a new field, right? So a lot of mechanisms need to be understood. I'm sure you agree with us that insights precede applications. You have to understand better for the mechanism, then you can do some translation, or I can say the application.
Thank you. Amanda, were you going to make a comment?
No, I was going to say this is sort of the perennial question at the NIH that we seem to grapple with, is the concept that, you know, there are some that don't believe that the reviewers accept basic biology, but, you know, we, as program officers, I will say that's not the case. There's no expectation that understanding the fundamentals of how something works is going to be able to lead to translation, though it might. And if you do put it in your application, then the reviewers are going to think hard about it. So make sure that you're not just using the translatability to try to sell the basic science, and the basic science can sell itself, right? There's no need to push it into the future, into the unknown, you know, sort of focus on the known.
Okay, I think that clarifies the issue. Paola Di Vieti asks, is the letter of intent required and binding?
Technically, no. It is helpful, however, because you will note that most RFAs, there's a letter of intent, and then and typically the letter of intent is like a month after the RFA is published, and then the due date is a month after that. We built in a lot of planning time here because the hope is that people will take the spirit of interorgan communication and tissue crosstalk seriously, because we take it seriously, and it does take planning to set up collaborations that haven't been ongoing, and so the RFA was published December-ish, and the letter of intent is April 1st, and then the due date is June 1st. So there, and the as you can imagine, if there are 20 different organ combinations, it's going to take some heroic effort by the SROs to come up with the scientific expertise to cover that. And so a letter of intent outlines for the SRO the sorts of expertise that they're going to need. And so a letter of intent is very helpful for plan. Even though, you know, it's 2 months between the letter of intent and the due date, you know, and then, but planning for the review starts early. And so a letter of intent is very helpful.
Excellent, thank you. Paul Cohen again asks, is there an interest in interorgan communication in age-associated disease, diabetes, cancer, cardiovascular disease, or is the focus on healthy aging?
It does not have to be healthy aging, no.
Okay.
But age. Age is an essential component of this. So AIDS in the context of aging.
Perfect, that makes it very clear. Marco Brotto will ask, will this U01, like in other cases, have a U34 implementation first?
No.
No, that's generally for clinical trials, just to make sure that you have clinical trial readiness. Not
That, okay.
Yeah.
No.
So it's not, the answer is no.
No.
Correct.
Okay. So I think that we don't have any more questions here, but in the meantime, we are almost reaching the end of the session. So if the panel would like to make a final advice for our attendees, a practical tip on how to get successful in this application, I will start with probably with Amanda.
I was hoping you would start with John. No, I mean, obviously, get in touch with us always earlier, right? So that we can help walk you through the expectations. We will talk to you about what we think is hitting the mark for the RFA early. So I think it's, as always, my first piece of advice at all stages of development of an application is please contact us, because we would like to help you refine it and answer any questions you have. We don't want any surprises as we get closer to the due date. I'll leave it at that.
Thank you very much. Uluwalem?
I will say contact the respective program officers. Send if you are planning to apply, send the specific aim page, and then set up a brief call or a meeting. So I have one RFA which posted last October, and it's going to, the due date is next month, February 12th. And I have spoken with several, prospective applicants, planning to apply for this RFA. So please do that. It will help whether it's in response to the RFA or not. The program officers will tell you that.
Thank you very much. Hongwei?
Yes, so first, any questions, just feel free to ask. And the second, I want to add, any model system are welcome, not only limited to the human subject, mouse, rats, any other organ are welcome. Thank you.
Thank you very much. John?
All of the above.
Oh, that's unusual.
So no, I think everybody is so. In my regular job, I read a lot of aims pages. So send an aims page if you have questions. I mean, we can better help you if you outline what you're trying to propose. And those of you that know me know that I will tell you exactly what I think. And you can agree or disagree. It is. I mean, I can advise, but basically you do not have to agree. But it is, the onus is on you to convince the reviewers that you can get the job done. And a lot of you have reviewed, so you know what that entails.
Excellent. Thank you very much. I really want, on behalf of the Gerontological Society of America and NIA, to thank the panel of speakers. I think it was very clear, very interesting. We had a very interactive discussion. I thank everyone for taking the time to attend, encourage everyone here to apply and build up a very nice story. Again, to the international attendees of this webinar, encourage everyone to contact your PIs or your collaborators in the States, and contact our health science administrators and discuss with them. You see, they are very accessible, they are very friendly, and I think that we can get a very, very interesting interaction and develop this field, that very interesting field of interorgan communication. With this, I also want to encourage all you guys to hopefully you will submit your papers that will come from this RFA or any other initiative or any project that you are running at this moment to our journal, the Journal of Gerontology Biological Sciences. We are very much interested on interorgan communication and aging. So you have data already, please feel free to send your results to us. With this, again, I would like to thank your attention. Before I would like to transfer to Jim to give the final words. And thank you, everyone. And I wish you a very nice afternoon. Jim?
Thank you all for your participation. I hope that this has been an informative and helpful webinar. Stay tuned for future webinars. And again, this will be recorded and available to you on the GSA website. Have a good day.
Bye. Take care.
Bye, everybody.
Have a good afternoon.